So, hello students, good afternoon all. So, let's start our class, and this is our very first class, and our first chapter is pharmaceutical analysis. Actually, this is a basic thing, so you have to understand it very clearly. First of all, we have to know what is pharmaceutical analysis. Actually, it is a branch of a chemistry, and it is a technique. It's a very important technique in order to identify a sample or a compound or a substance by using different methods like manual method, chemical method, instrumental method, etc. Manual method means by we are only doing. Then chemical methods means titration. It's a good example for chemical method. Instrumental method means using instruments. For example, UV instrument or mass spectroscopy, etc. Then, different techniques of analysis. So, there are two types, that is qualitative analysis and quantitative analysis. Qualitative analysis means we are identifying a sample by means of color, taste, viscosity, solubility, etc. Then quantitative analysis. Quantitative analysis means we are identifying a sample in the terms of numerical values. That means, for example, we are doing it by different methods like chemical method, instrumental method. For example, we are doing a titration. So after doing titration, we'll get some values. So using that, we are calculating about the sample. So that is called a quantitative analysis. So in chemical methods, there are three types. One is titrimetric analysis, gravimetric, gasometric. Second method is physicochemical methods. Third one, microbiological methods. And finally, biological methods. First one, that is titrimetric methods. That is, or we can say volumetric methods. It is nothing, it is titration. You have already done it in your school sessions. So we'll get very accurate and precise measurement using titration. How we are doing titration? We are interacting the solutions with reagent. And there are many examples for this volumetric analysis like acid base titrations, non aqueous titrations, redox titrations, precipitation titrations, complexometric titrations. So we'll study in our upcoming chapters about these titrations detail. Now you just memorize these names. Gravimetric analysis. Gravimetric analysis means we are converting an element or a radical into pure stable compound. So after converting into a compound, then we used to wait and we will do our experiment. So that is called a gravimetric analysis. So what is the difference between this volumetric and gravimetric analysis? In volumetric analysis, it is very rapid method, means fast method. Gravimetric means it is very lengthy and it is time consuming also. And more accuracy is for gravimetric analysis. And titrimetric means it is very specific and versatile method. And gravimetric means we are converting a compound or an analyte into a precipitate. That is called a gravimetric. Then gasometric. Gasometric means volume of gases, measuring volume of gases. So many gases like cyclopropane, carbon dioxide, nitrous oxide, oxygen, etc. can be determined by this method. Actually, we used to measure gases by gas burettes or nitrometers. Next one, instrumental methods. Instrumental methods means it is very selective and it is very sensitive methods while comparing with the other methods. And we are measuring a physical property or a chemical property by this method. That means with this method we can determine current potential, electrical conductivity, optical density, specific rotation, refractive index, etc. by different instruments. And let's see some features of the instrumental methods. As we told that they are very selective and they are very sensitive also, more expensive also, and they are very rapid, very fast, and less time consuming, and it is fully automatized and computerized, so we can repeat it many times our experiments. Example for the physical properties. So by instrumental methods we can determine all these properties. Example: electrical potential, conductance, radiation, thermal properties, potentiometry, conductometry, polarography, spectrophotometry, thermal method, etc. Next, microbiological methods. Microbiological methods, it is a comparison actually. So there will be two samples, one test sample and one is standard sample. And we are measuring the concentration of the antibiotic in it, or a growth of bacteria in it. That is called a microbiological method. So we usually go through mainly two methods, that is cup plate method and tube assay method. Next is biological methods. Biological methods means when a potency of drug or a derivative cannot be properly determined by physical methods or chemical methods, then only we will move to biological methods. That is, we are observing the biological effect of that sample. That is called a biological method. Then, certain definitions. Solution. Solution means it is a mixture of two substances. That is, solute is dissolving another substance. That is known as solvent. Then, standard solution. Standard solution means we know the strength of that particular solution. That is called a standard solution. Titration means that is we are adding solution from burette to conical flask, and reaction, the reaction will undergo, that is called a titration. Then titrant. Titrant means the solution in burette, that is called titrant. Titrate. Titrate means the active substance present in the solution, that will be in conical flask. Equivalence point and end point. Equivalence point means the point at which the reaction just complete, that is called equivalence point. Then end point means at the point at which the color changes, that is called an end point. Then indicator. Indicator means it is a reagent to identify a titration is complete. Then titration error. Titration error means in case of titration there will be end point, there will be equivalence point. That is theoretically and practically. Sometimes they will be same, sometimes they will be different, slightly different. So that difference is known as titration error. So that's for today. Thank you.